Yeah. Good morning, Sunita. I hope you're following the recorded. I think you missed a few classes now. So please kindly go through the recorded in case if you miss anything, okay? Don't miss. Okay. Good morning, Akshay. Good morning. Okay, what about others? Okay, no problem. So let's continue. So friends, in the yesterday class, we were started discussing about the cell communication. In the cell communication, we are discussing first different type of CAM molecules, is it? And we understand that there are four major CAM molecules are there. Good morning, Shafia. That is the cadherin, selectin, Ig superfamily, integrin, is it? So in the yesterday class, we understand cadherin. Good morning, Saima. We understand cadherin, we understand selectin, we understand the integrin, and we are left with. Good morning, Sumaya. We are left with one more. What we can call the CAM molecule, that is called as a immunoglobulin superfamily CAM molecule. Friends, all of you please listen very carefully. Immunoglobulin means it is not an antibody, it's a type of CAM molecule only. But what is the point you people has to remember? Its a structure resembles of immunoglobulin or antibody. That is the only reason, friends, we are calling it as the immunoglobulin superfamily CAM molecule. And please keep it in your mind. Of course, friends, this immunoglobulin superfamily molecule will show homophilic interactions. Kindly do not get confused with this particular point, which means, good morning, Brunalini. This particular immunoglobulin will go and associated with the another immunoglobulin CAM molecule present on the same group of the cell. That is the reason we can say they are following the homophilic interactions. Of course, friends, if you look into the structure, the structure is the common. What is that? Long extracellular domain, a single span, only one time it can transpan the plasma membrane. And friends, a short cytosolic domain. Kindly do not get confused with this. And friends, listen to me very carefully. This is called as what, friends? Extracellular domain. Undoubtedly, friends, extracellular domain of one Ig superfamily CAM molecule, good morning, Priya, will be connected to the extracellular domain of the another immunoglobulin superfamily CAM molecule. And keep it in your mind, the transmembrane domain and the cytosolic domain never resemble like immunoglobulin. Listen to me carefully. Cytosolic domain and the transmembrane domain never resembles like immunoglobulin. Only, friends, a part of the extracellular domain. Listen to me carefully. The part of the extracellular domain exactly resembles like immunoglobulin. That's why we are calling it as the immunoglobulin superfamily CAM molecules. Friends, if you look into our body, I am talking about the human, we are having, friends, different type of immunoglobulin superfamily CAM molecules are there. Okay, now the point is here, based on what we categorized them into different type of immunoglobulin supramolecule. Everyone remember, friends, based on the tissue where they are getting expressed actually. Very simple point. Based on the tissue wherever it is expressing. And friends, you people no need to remember the CD molecules, don't worry at all, and remember whatever I am saying. There are many type of CAM molecules are there. So listen to me. Intercellular adhesion CAM molecule 1, ICAM1. Intercellular adhesion molecule 2, ICAM2. Then, friends, VCAM1. Vascular cell adhesion molecule. Always remember the short names. By that you can remember it for the long time. PECAM. PECAM1, platelet endothelial cell adhesion molecule. These are all names were given based on which cells they are basically getting expressed. That's all. Then friends, MADCAM, mucosal addressing cell adhesion molecule. Do not get confused with that. Then friends, followed by the NCAM, neural cell adhesion molecule. Always remember this particular point. Listen my statements very carefully. These are all the different type of immunoglobulin superfamily CAM molecules present in our body, which were named based on the tissue wherever they are expressing generally. Kindly do not get confused with this particular point. And friends, in the diagram, you all people can understand one point very commonly, that is the extracellular domain of the immunoglobulin associated with the each other. And please listen friends, this is the cytosolic domain, right? Keep it in your mind. The cytosolic domain of the immunoglobulin superfamily molecule, basically you all people can understand in this picture very clearly. You can see here they will be associated with the actin filaments. We all know that they will be associated with the actin filaments with the help of a linker molecule, or listen friends, with the help of a adapter molecule. What are this linker or adapter molecule? You have to remember, we can call it, now we are going to discuss this, ankyrin and spectrin. Ankyrin and spectrin are the major linker or adapter molecule through which this particular immunoglobulin superfamily CAM molecules will be associated through the to the cytoskeletal elements such as the actin. Kindly do not get confused here, and please try to remember maximum amount of the information. Okay, so friends, so far what we have studied is the type of CAM molecule. Once again I am saying this topic is a very rigid topic with very great patience. You have to read, you have to remember, because it is extremely important, even though it's a very rigid concept. Okay, so far we have studied different type of CAM molecule, especially we have studied 4 type of CAM molecule, is it? So next, before going to the extracellular matrix proteins, we have to read compulsory different type of junctions. When I use a word junction, where the junction will be present, friends, everyone listen, the junction can be present between cell to cell, or the junction can be present between cell to the extracellular matrix also. See, how did we categorize the junctions? Once again, the point is very clear, based on their arrangement. What friends? The simple parameter. Based on their arrangement, we categorized the junctions into different types, based on between the cells or between the cell to matrix. So these are all cell to cell interaction. You all people kindly look into the diagram very carefully, friends. I will explain one by one very elaboratively. Tight junction present between the two cells. Adherence junction present between the two cells. Desmosomes present between the two cells. Undoubtedly gap junctions also present between the two cells. You all people look into this. Hemidesmosomes, which are a part of the desmosome only, structurally and functionally 90% similar to the desmosomes only, but there is a slight difference. We will see that. Hemidesmosome and focal adhesion, they are not connecting cell to cell, friends. They are majorly connecting the cell to matrix. Kindly do not get confused with this particular statements. Now we will discuss one by one junction very elaboratively. In every junction, you people has to remember structure, function, most importantly, what kind of proteins that are involved in that particular junction formation. These are all the only questions, friends, we can expect in the exam, in the majorly in the 4-marker session only. Okay, first let me give you the overview of the functions of the cell junctions. Later I will explain you very briefly and elaboratively about what we can say that one by one junction. Everyone look into this tight junction. Friends, one point you all people has to remember, very important point. When I use a word tight junction, you always keep it in your mind. Tight junction can yellow liquid molecules easily. No. Why I am highlighting this point is that, see, this is one cell, okay, and this is the another cell. The tight junction seals, everyone listen, the tight junction seals the two cells in such a way that even it never ever allow the movement of liquid molecules also. That much tightly it will seal the two cells. Kindly do not get confused with that particular point. Next one, friends, adherence junction. Name itself indicating na adherence, attachment. Just listen my statements very carefully. Here you are able to see red green color ones. These are all what? Adherence junction. Listen. What is the simple function of the adherence junction? Focus. You are able to see, right, this red color bundle. This is called one actin bundle. So adherence junctions are those type of junctions, friends, which are joining actin bundle in one cell to a similar actin bundle in the neighboring cell. That is what exactly, friends, we can call the adherence junction. Kindly do not get confused with this. So friends, adherence junction are joining the actin bundle. Friends, if you look into the desmosomes, what you have to remember, listen to me. Desmosomes joins the intermediate filaments in one cell to those of in the neighbor. That is what exactly, friends, we will be calling it as the desmosomes. Do not get confused with that. Remember my point. Desmosomes connecting the intermediate filaments in one cell to the intermediate filament of another cell. And friends, when you look into the hemidesmosome, hemidesmosomes are also involved in the joining of intermediate filaments only. There is no doubt about it. And they are joining the intermediate filaments not from cell to cell. They are joining the intermediate filaments from cell to the basal lamina. Kindly do not get confused with this particular point at all. Okay, now you listen my statements very carefully. The last and final one, gap junction. Name itself indicating a gap, which means it is yellowing the passage of molecule. Friends, please remember, when we use a word gap junction, it doesn't mean that it is yellowing the every single ion and every single molecule. No. Any kind of a ion or molecule has to be transported through gap junction. It should have a particular size. If that particular ion or a molecule beyond that particular size, it cannot be transported. We're gonna discuss it very elaboratively. Okay, let's start with the first one, the most important one, tight junctions. Okay, listen everyone very carefully. I want you people to remember the other names, especially the tight junctions. We widely call it as, friends, zonula occludens. This is one cell, this is the another cell. The two cells are sealed on epithelial sheet. What, friends? The two cells are sealed on an epithelial sheet, and finally they are going to form, everyone listen, virtually impermeable barrier. They are impermeable in such a way that not only the solid molecule, even any fluid molecules also, it never allows. So that much tightly it associated with the each cell. And remember, friends, the first we have discovered the tight junctions in the intestine, stomach lumen, even some of the blood cells. I want every student has to remember one important point. Please do not consider that the same type of tight junctions will be present in every single cell. No, not. In different cells, friends, different types of tight junctions will be present. But The tight junctions will be made up of the common proteins. I will tell you in the next slide. You all people look into this. Cell 1, cell 2, cell 3, cell 4. So cell 1 and cell 2 are tightly associated with each other with the help of a tight junction, which never ever allow movement of any molecule from one cell to the another cell. Now, friends, all of you please kindly look into the diagram. This picture will help us to understand very nicely. See here, this is the electron microscopic picture, the same picture. See here, this is one cell, this is another cell. One cell, another cell, one cell, another cell, one cell, another cell. The two cells are brought together. Everyone listen. The two cells are brought together and they are very closely packed with each other. The two cells are brought very close together and they are very tightly packed with each other. That is how the tight junctions will be associated. Okay, what are the various type of proteins that are composing the tight junction? Everyone listen, friends. The tight junction is majorly made up of 4 proteins. 4 proteins are also very important proteins. What are those, friends? Listen, you have to remember the properties of all the proteins. It's a kind of rigid topic, pay attention. The tight junction is made up of occludins, claudins, tricellins, then followed by another protein called as JAM protein. Kindly do not get confused with this. And friends, what is the point you have to remember? Except JAM protein, occludins, claudins and tricellin, they are all can transpan the membrane. How many times, friends? They are transpanning the membrane tetraspan, nothing but 4 times it is transpanning the membrane. And friends, if you look into the JAM protein, everyone keep it in your mind. JAM protein, it is the single transmembrane, only single transpan. It can move the membrane only one time. All the students look into the picture very carefully, friends. Claudin, occludin, tricellin. These 3 are transpanning the membrane 4 times. You can understand it very clearly. And you can see na JAM, it is only transpanning 1 time. Now here comes very important point. I was repeatedly saying to all of you that the tight junction associated with between the 2 cell and it will seal the 2 cells very tightly. Now the point is very clear. Listen to me. This is cell 1, cell 2. On the cell 1 tight junction is there. Correct na? On the cell 2 also tight junction is there. There is no doubt about it. But the tight junction present in the cell 1 has to be associated with the tight junction present in the cell 2. Yes or no? Of course yes. But how? That is the point. With the help of a JAM protein only. So you know what is the line I can give you? JAM protein can act like a better adhesion molecule. Don't get confused, friends. JAM protein can act like a better adhesion molecule which join the tight junction present in one cell to the tight junction present in the present on the another cell. By that, the 2 tight junctions of the 2 cells can be associated with each other very strongly. Not only that, you all people has to remember that the tight junctions can also associated with many kind of a cytoskeletal proteins. What kind of cytoskeletal proteins majorly tight junctions will be associated? Of course, with the actin filaments. Everyone listen. If the tight junctions wants to be associated with the actin filaments, they will be requiring many kind of a adapter proteins as well as, friends, scaffolding proteins. The point is very simple. With the help of scaffolding and adapter proteins or the connector molecule, the tight junctions can be associated with the actin filaments which are present in the tight junction. Okay, and I just said that there are four proteins are there: claudin, occludin, tricellin and JAM. And just now we have discussed very clearly the major function of the JAM is that which is connecting the tight junction of the one cell to the tight junction of the another cell. That point is very clear. Now occludin, claudin, tricellin, among all, which is the most important protein of the tight junction? This also you can get in the part B or as one of the statement in the part C. Always remember friends, among all, the claudin is said to be the most important protein of the tight junction, because friends, all the students listen, 90% of the functions performed by the tight junction which is exclusively mediated by the claudin molecule only. Do not forget that. That's why friends, claudin is said to be the most important molecule. Please do not forget this. Okay, now I told you now friends, you people do not confuse. An example, let's take a kidney, let's take intestine. In the kidney also tight junction is present, in the intestine also tight junction is present. So in the tight junction, which is the major protein, just now we said claudin, so here also claudin is present. Now you should not think that both claudins are same. No, there will be a specific type of claudin will be present in every single tissue. So in kidney there is a one specific claudin is present. In the intestinal tight junction also there is another specific claudin is present. Kindly do not get confused with that particular point. Listen friends, a specific claudin found in the kidney epithelial cell. For example, magnesium is extremely important and it has to be reabsorbed from the urine into the blood. An example, all the students listen to me. We should not allow, friends, focus what I am talking. We should not allow the magnesium to be reabsorbed from the urine into the blood, which means we should not allow the magnesium to be excreted in the form of a urine. We require magnesium for certain essential functions, so we have to be reabsorbed. So we have to block. What we have to block? We have to block the movement of the magnesium. So how can we block? Undoubtedly friends, we can block with the help of a tight junction only. So with the help of a specific claudin, that claudin only found in the kidney epithelium, that is specific, that is extremely responsible for magnesium reabsorption only. Kindly do not get confused with this particular point at all. Next one is second major transmembrane protein in the tight junction is, see, I told you the major one is the claudin, then occludin and tricellin. Friends, till today we did not understand the clear functions of the occludin and tricellin, but you know, available data is indicating that occludin and tricellins will be responsible for the stability of the claudin, which is the major protein of the tight junction. Do not get confused with that. Occludin is the major protein and claudin is the major protein. Occludin and tricellin we really don't know, but the available data or available research suggesting that occludin and tricellin may be involved in the stability of the claudin. And I already told you friends about the JAM protein. With the help of JAM protein only, tight junction in one cell can be connected to the tight junction in the another cell. In one simple line, what you can understand, JAM proteins are the one friends which plays a key role in the homophilic adhesion. We know, right, very clearly homophilic adhesion means the JAM protein is going to be associated with the another JAM protein present on the other cell. Remember and don't forget this. That is about the first junctions, tight junctions. Do not get confused with this. And where the tight junction is present, tight junction is not present between cell to matrix. The tight junction is present always between cell to cell, right? Now friends, now next what we are going to read is the anchoring junction. Friends, all the students keep it in your mind. The moment, understand, please remember the statements. You never get any experimental question, any graphical, any analytical. You will get only one way, either statement or matching. That's all the question. Okay, but very straightforward question only you will get from truly from statements. Okay, when you heard the name of a anchoring junctions, I want you to remember a common point. Anchoring junctions are the one which plays a key role in providing the strength. At the same time, strength means it is the one which will provide the mechanical attachment. Kindly do not get confused with this particular point. Mechanical attachment can be mediated by this particular anchoring junctions. And friends, we just read, right, the tight junctions are present between cell to cell. But friends, when you look into the anchoring junctions, what is the point you have to remember? Anchoring junctions are present between cell to cell also. At the same time, you all people look into this. This is one anchoring junction. One second, look into this. This is an anchoring junction present between two cells. Not only cell, it is present between cell to matrix also. So tight junctions are present only between cell to cell. The anchoring junctions are present between cell to matrix. So friends, do not get confused. Listen my statements without confusion. Here you will get lot of confusion. The first point, anchoring junctions providing the strength and acting like a mechanical attachment. Anchoring junctions are the one which are responsible for the structural integrity of the tissue. They are present between the cell to cell and cell to matrix. Now the point is here, the anchoring junctions basically made up of two type of proteins. Do not get confused with my words. Anchoring junctions are composed of two different type of proteins. Intracellular anchor proteins, transmembrane adhesion protein. Name itself clearly suggesting us, right, intracellular anchor proteins means which are present in the cytosolic domains. Transmembrane protein which are basically transpanning the plasma membrane. Now I want every student has to listen to me carefully without confusion. Every anchoring junction made up of two type of proteins. Based on these proteins means what? Based on the composition, based on the function performed by them, anchoring junctions are divided or differentiated into two types. Do not get confused. You people has to remember compulsory. The first type is the adherence junctions and desmosome. It is the first group of the adherence junction. First group of the anchoring junction. Do not confuse and do not forget. Next type, focal adhesion and hemidesmosome. These are all the second type of the anchoring junction. And I will tell you in a simple points. Adherence junction and desmosomes, which are the first type of the anchoring junction, they are majorly responsible for cell to cell interaction, and they are always associated with the transmembrane cadherin family. I told you, no, every anchoring junction made up of transmembrane protein, intracellular protein. Now what is the hint I am giving to you is to remember easily. Adherence junction and desmosomes are associated with each other with the help of a transmembrane protein, that is the cadherin. Focal adhesion and hemidesmosomes are the second type of the anchoring junction. They are exclusively present between the cell to matrix, and they are also having a transmembrane protein. There is no doubt about it, and that transmembrane protein is majorly of the integrin. Do not confuse. I'll re-explain in 1 minute. Focus. We read the tight junction. The second junction is the anchoring junction. The anchoring junction present between cell to cell also, even cell to matrix also. It is responsible basically for the structural integrity of the tissue. And every anchoring junction made up of two proteins which we can call intracellular and transmembrane protein, and these anchoring junctions are broadly categorized into two types based on their composition and majorly based on the function performed by them. Such a simple step it is. Adherence junction and desmosomes present between cell to cell. These two having the transmembrane protein as the cadherin. Never ever forget this statement. Focal adhesion and hemidesmosomes are the second type of the anchoring junctions. They are present between the cell to matrix and they are having the transmembrane protein as the integrin. Do not forget the statements. Okay, now let's talk about one by one. So what is the first type of the anchoring junction? Adherence junctions and desmosomes. So let's understand about the adherence junction and desmosome. Friends, kindly do not get confused. You remember? Tight junctions. What do we call it, the tight junctions? We call the tight junctions as the zonula occludens. Correct na? And friends, what do you call adherence junctions here? All the students keep it in your mind. Adherence junctions. We will be also called as a zona adherence. What is the first statement? Every student has to keep it in your mind when you are reading the adherence junction. The first statement you have to keep it in your mind. It is responsible for cell to cell attachment and it is having a transmembrane domain of cadherin. Look into this, look into this. This is one cadherin. 1 second. Okay, look into this. This entire molecule is cadherins. Okay, this is cell 1, cell 2. So one cadherin present in one cell can be associated with the another cadherin. Correct na? So understand friends, this entire part we can call adherence junction. In the adherence junction, what is the function of a cadherin? Cadherin is the one which is acting as a transmembrane domain. With the help of transmembrane domain only, two cells can be associated with each other. Not only connecting to the external environment, you all people can also understand. They are connecting to the cytosol also. You all people can see, right? These are all the actin filaments. Now friends, if this particular cadherin molecule want to be associated with the cytoskeletal domain, undoubtedly what do they require? They require compulsory linker molecule. I told you, right? Linker molecule or adapter molecule. What are that linker or adapter? We can call the catenins. We can call the vinculins. We're gonna read it in the upcoming slide, but it is very important, friends. You people has to remember the name of the linker or adapter molecule compulsory. Alpha catenin, beta catenin, vinculin are the type of the molecules which are connecting this cytosolic domains to the actin filaments. Kindly do not get confused with this particular point. And friends, where the adherence junctions will be present when we are reading the tight junctions, I have given you examples. That's all. Na? It is present in the kidney epithelial cells. You have to remember tight junctions are present in every single cell. Similarly, adherence junctions are also found in variety sites within the body. Many sites. The point is very clearly we studied. The cells at adherence junctions are held together by what molecules? Cadherin molecules. At the same time, I have clearly mentioned to you, cytoplasmic domain of the cadherins is linked to the actin filaments. How? By a linker or adapter proteins called as catenins. Kindly do not get confused with this particular point. And friends, I want you to look into this beautiful diagrams. This is the cadherin. You can see now, It is present, this is one cell, this is the another cell, and two cadherin molecules associated in between what? This is the what we can say that this is the area where the movement of molecules occur, and this entire area, what do we call it? Good morning Arrow. This entire area we can call it as anchoring junction. Majorly they contain transmembrane domains of the cadherin molecules. And friends, I hope you are all recalling this diagram of invagination. While reading the process of gastrulation in the developmental biology, we have studied invagination, involution, epiboly, convergent extension, delamination, ingression. I hope you all people remember. Now you see here, this is how the epithelial sheet is there. This is how the sheet is there. Now what is happening? You all people know, right? There will be a change in the size. Correct na? Good morning. There will be a change in the shape. Because of the change in the size and the change in the shape of the epithelial sheet. See friends, it is undergoing a kind of invagination. Because of invagination, what is happening? You can see here, it is forming a epithelial tube-like structure. See, originally it is a large sheet. You all people can observe. This large sheet, now it is converted into a small tube-like structure. How it is possible? Because of the mechanism of invagination. In that invagination, which molecules are involved, friends? Of course, this particular anchoring junctions which contain cadherins are involved. So, anchoring junctions are of two types. One we can call the adherence junction and desmosome. We studied the adherence junction. Now we are going to study the desmosomes. And friends, the moment you heard the name of a desmosomes, I told you now friends, remembering the other names only very important, because they most of the time they will, to make us confused, they'll give the other names instead of the general names. Tight junctions we can call zonula occludens. Adherence junctions we can call zona adherence. And friends, most importantly, desmosomes we will be calling it as the macula adherence. Kindly do not get confused with this. And one more important point, friends, when you heard the name of a desmosome, just keep it in your mind. Desmosomes are a button-like structures. Okay, what friends? The desmosomes are button-like structure. All the students, please look into the diagram very carefully, friends. Here the red color you can see, right? That is only desmosome. Correct na? Now you all people understand without confusion, friends. Here you can see now blue color. They are all what? Keratin filaments. Okay, and these are also keratin filaments. You want to understand very clearly. This desmosomes, listen friends, connecting keratin filaments of one cells to the keratin filament of the another cell, correct na, via cadherin as their transmembrane protein. You can see very clearly in the diagram, because we read it clearly. Adherence junction and desmosome, they will use the transmembrane adhesion proteins as cadherins only, is it? Now do not confuse at all. So desmosome connecting keratin filament of one cell to the keratin filament of the another cell with the help of a transmembrane domain, that is the cadherin. Now once again, don't confuse here, listen to me carefully. I said to you that anchoring junction is made up of two type of proteins, correct na? So desmosome is also what? A type of anchoring junction only. Hope you are getting me very correctly. Desmosome is also a anchoring junction, so it is also made up of a two proteins, correct na? What are those two proteins? Transmembrane protein as well as the intracellular protein. I want you to remember without fail all these proteins. What are the intracellular protein? Please keep it in your mind. plakoglobin and desmoplakin. You have to remember for the exam. Plakoglobin and desmoplakin. These two are the intracellular protein. Undoubtedly, friends, it is associated with the transmembrane also. Correct na? Transmembrane nothing but cadherin. What are the examples here? The examples are desmoglin. You have to remember, there is no shortcut here. And desmocolin. Desmoglin and desmocolin. These two are basically belongs to the family of cadherin only. Please kindly don't remember as cadherin family. Remember what are the two proteins that belongs to the cadherin family. Remember that. Desmoglin and desmocolin, which are transmembrane, and intracellular is the plakoglobin and desmoplakin. Okay, so this is what first type of the anchoring junction. What is the first type of anchoring junction? Adherence junction and desmosomes. And what is the second type of the anchoring junction? Focal adhesion and hemidesmosome. You don't need to read much about the focal adhesion, it's not greatly important. Majorly focus on the hemidesmosomes. And friends, whether it is the focal adhesion or the hemidesmosome, what did I tell to all of you very clearly? They are majorly present between cell to matrix. And listen to me very carefully. Here we have written that hemidesmosomes are half desmosome. I won't call actually half, I will call 90% actually. 90%, friends, hemidesmosomes structurally, I am talking 90%, structurally and functionally similar. Desmosomes are also connecting the intermediate filaments of one cell to the another cell. What the hemidesmosomes are doing, they are also connecting the intermediate filaments only, friends. There is no doubt about it. But they are connecting the intermediate filaments of one cell, not to the other cell, one cell to the matrix. All the students listen to me very carefully, friends. This is the hemidesmosome. What? These are all the hemidesmosomes. What they are doing? Everyone listen. They are connecting this particular keratin filaments or the intermediate filaments to the basal lamina or to the extracellular matrix, the yellow color one which you can see here. So the only difference is cell to matrix, whereas desmosome is cell to cell. And what is the another very unique statement you have to remember? No, desmoplakins and desmoglobins are not adapters, not at all. No dear, I am telling you now here, intracellular proteins, that's all. They are present in the cytosol or cytosolic domain, that's all. But they are not actually what we can say that adapter protein. There are certain adapter proteins are there, and Sumaya, you can see in this diagram the light orange color. These are all the adapter proteins. Adapter proteins are same dear, catenins or the vinculins. Okay, I got your doubt. Plakoglobin and desmoplakin, actually they are associated with the intermediate filaments and actin filaments, but they are not truly linker proteins. Okay, they are just arranged in the intracellularly, that's all. Their arrangement is intracellular, but they are not a linker or adapter protein. Okay, yeah, so I was saying that, listen to me, adherence junctions and desmosomes both are associated with each other with the help of a transmembrane domain, cadherin. And what is the statement I told you to remember without fail? Focal adhesions and hemidesmosomes are associated with each other with the help of a transmembrane. It consists of integrin. Do not confuse and do not forget. And I want every student has to look into this beautiful diagram. Listen my statements carefully, all of you. This is, which are I am marking with the red color, this is hemidesmosome. You all people see this black color ones, these are all the intermediate filaments. What did I tell to all of you? The hemidesmosomes connecting the intermediate filaments to the ECM, extracellular matrix. We all people know, we are going to read in the next upcoming slides. The ECM consists of what? Collagen, correct na? Fibroin, nidogen, we will read it, elastin, proteoglycans, many proteins are there. But how? All the students look into the picture very carefully. With the help of integrin proteins. With the help of integrin, you can see the green color, and Simaya, you can see here little more clearly. These are all the anchoring proteins. These are all the adapter proteins. Okay. Now you look into this. These are all what, friends? Do not get confused here. This is what? Integrin, because we read na, on the transmembrane domain there is a presence of the integrin. With the help of integrin, this hemidesmosome connecting the intermediate filament to the extracellular matrix proteins. Do not get confused. Okay. During the revision time, you people no need to read all this and no need to waste the time. To save the time, what you do is you majorly focus on this particular table. In the table it was very clearly written which are adapter proteins, which are intracellular proteins, which are transmembrane adhesion protein. And if you can remember the extracellular ligand, remember, otherwise leave it, there is no problem at all. But majorly focus on transmembrane adhesion protein, intracellular cytoskeletal protein, and intracellular adapter protein. Just now we read, na friends, adherence and desmosomes are cell to cell. We clearly discussed. Adherence junction consists of intracellular what? Catenin, beta, alpha catenin, beta catenin. Undoubtedly they are associating with which cytoskeletal element? We read it, actin filament. Desmosomes we read, it contains plakoglobin, desmoplakin. They are associating with what? Intermediate filament. That's all. And these are all what? Focal adhesions and hemidesmosome. If you don't need to remember the focal adhesion, majorly focus on the hemidesmosome. Hemidesmosomes connected to the intermediate filaments with the help of a adapter protein called as plectin. Adapter is the plectin, but what is the transmembrane protein? Transmembrane protein is the integrin. Kindly do not get confused. Only read the table during the time of revision. So we read tight junctions. We read anchoring junctions. In the anchoring junctions, we read desmosome, hemidesmosome, as well as the adherence junctions. Now the next type of the junction we can call communicating junction. The name itself very clearly indicating communicating, because it is allowing the movement of the substances. It is involved in the intercellular exchange. Not very large molecules like proteins. No, proteins never be transported greatly. Okay, small molecules like small ions and small molecules only, which are having low molecular size and weight. Weight also important. They will be transported from one cell to the another cell. What are the best examples of the communicating junctions? The best examples are, friends, gap junction as well as the chemical synapse. Kindly do not get confused. It's very important, friends. Both are extremely important. Focus. Gap junctions. Once again, I am telling to all of you the name of the gap junction, or listen to me. Gap junction made up of a polymeric protein. Do not confused. The gap junction made up of a polymeric protein called as connexons. Connexon is the major protein of the gap junction. How does the connexon will be formed? Connexon is a polymer, right? So connexon is made up of monomer. How many monomers will come? 6 monomer. What do you call every single monomer? Connexin. 6 connexins molecule. 6 connexins molecule come close together and they can form one connexon. All the students look into the diagram very carefully. This is one cell. This is the second cell. Correct na? Now the two cells are associated with the gap junction. What is that called as? Connexon. You can look into the diagram. This is the connexon. How does this connexon is made up of? This connexon is made up of 6 connexin molecules. Do not get confused with this. You know friends, when you look into the structure of a gap junction, we will see that the two cells are associated very close to each other, but not actually. All the students will understand. The plasma membrane comes in very close contact to one another. But friends, approximately there is a 3 nanometer distance is there between the two plasma membrane of two cells. So friends, at that 3 nanometer gap only, these gap junctions will be spanned. The gap junctions will be arranged. So the gap junctions were first discovered in the myocardium of the heart, because you know, right, in the heart electrical transmission has to occur continuously. What is electrical transmission? Movement of the ions or movement of the molecule. It has to move compulsory, correct na? So how does this ions are moving? Of course with the help of gap junctions only. What kind of ions or molecule can be easily transported through gap junction? Inorganic ions, sugars, amino acid, nucleotides, vitamins, and one of the most important point, most of the secondary messengers like IP3, like cyclic AMP, these are all transported through this particular gap junction. But none of the macromolecule like polysaccharide, like lipid, like protein, they cannot be transported. Only the molecules which are up to 1000 kilodalton molecular weight, only they can be transported. You can see the picture very clearly, arrow. If it is more than 1000 kilodalton, they never ever transport through this particular gap junctions. Okay. So remember till this particular slide. In the tomorrow class we will start from again this particular slide of gap junction, and in the tomorrow class I'll finish the complete lesson. Okay, clear everyone? I hope it's clear. You can ask me if you have any kind of a doubts. Please join at 8:30 AM. We will continue the developmental biology. Okay friends, that's all for now. If you have any kind of a doubt, please ask me. Okay, perfect. I'll see you at sharp 8:30 AM. Okay, thank you all.